Hello everyone, my name is Tyler Posey and I'm a junior chemistry major in the Randall Research Scholars Program. This semester I've had the wonderful opportunity of working with Dr Meenakshi Arora at the Department of Translational Science and Medicine, and we've been working on oral delivery of paclitaxel using Squalene-Ligand conjugated receptor-mediated delivery. Now before I delve into this presentation tonight, I'd like to give a brief overview of what I'm going to be discussing. First and foremost, I'm going to share a short background on the field of drug delivery and using nanoparticles to improve drug delivery. I'm then going to go into the methodology regarding how we formulate our own squalene nanoparticles with ligands. After that, I'm going to share some of our preliminary results and how they reflect that our squalene nanoparticles will be an effective tool in improving the delivery of oral medicine. Lastly, I'm going to share what we hope to accomplish in the future with our research. So starting with the background, around the 1990s, scientists and doctors classified oral medicine into 4 categories depending on their permeability and solubility in the body. These are reflected in Figure 1, and I'd like to bring your attention to Class 4, which is shown in red. These drugs include our model, paclitaxel, which is an anti-cancer drug, and they have low permeability and low solubility. This means that they have a difficult time crossing the intestinal barrier to get to the systemic pathways, and they have an even harder time moving in those pathways to where they need to go. Now, as shown in Figure 2, although most current drugs belong to Class 1, which have high permeability and solubility, it's predicted that 70% of new drug discoveries will fall within Class 4. This means that we won't have a way to make use of these drugs without some sort of drug delivery system. Now, one of these drug delivery systems is using soluble nanoparticles to encase the insoluble drug that can cross the intestinal barrier and circulate the drug to wherever it needs to go. Furthermore, after COVID-19, the use of lipid nanoemulsions has become a more effective means of delivering drugs. It's also become a much more popular and conventional method. And of all the different molecules that are used to create these lipid nanoparticles, squalene has been extensively used due to the fact that it's readily found in nature, existing in shark liver oil and olive oil. Squalene has also seen use in treating our target disease, which is cancer, and it's already FDA approved. And its molecular structure is circled here. Now, though squalene has seen heavy use in creating nanoparticles to improve drug delivery, it's far from perfect, and that's for 2 main reasons. First, squalene is a very long molecule, meaning that it forms very unstable and loose nanoparticles. This means that it has a low drug loading capacity. Secondly, squalene nanoparticles, when they're unmodified, cannot perform targeted drug delivery. This means that squalene will cause off-site toxicity. So the question is, how can we counteract these flaws of squalene to maximize its use in designing nanoparticles to improve drug delivery? And our lab believes that the answer lies in attaching ligands to the squalene. Ligands, as shown in Figure 4, are molecules that bind to specific protein receptors in the body and have a unique biological response. Therefore, by attaching ligands to our squalene nanomolecules, we can not only reinforce their structural integrity, increasing their drug loading capacity in doing so, but we can also equip them with the ability to perform targeted drug delivery. This will increase the bioavailability of the drug. Now, moving on to the methodology. In order to create our squalene nanoparticles, we use a 2-phase process. In the synthesis phase, we create squalene molecules with ligands attached to them using a 5-step process. In the formulation phase, we combine our squalene molecules with ligands into nanoparticles with ligands displayed on the surface using emulsions. Starting with the synthesis phase, to attach our ligands to squalene, it needs to have a carboxyl group, which is circled. However, squalene in its normal form doesn't have a carboxyl group. In other words, it lacks functionality. So we need to add functionality using a method called squalenation, developed by Patrick Hoover, who did extensive work with squalene. Squalenation is accomplished by first reacting the terminal double bond of squalene with NBS and water to give a tertiary alcohol. We can then react this tertiary alcohol with potassium carbonate to get an epoxide functional group. In step 3, we can react this epoxide functional group with periodic acid. This gives us access to an aldehyde, which we can then oxidize to our desired carboxylic acid in step 4. Once we've received our desired carboxylic acid, we can react our squalene molecule with naringenin, our ligand of choice, to give our squalene molecule with a ligand as shown in the box. Now, after we have our squalene molecule with a ligand attached, we can create our nanoparticles using a 6-step emulsion process. This process begins by first preparing our organic phase, which consists of our squalene molecule with a ligand attached, our drug of choice, which is paclitaxel, and ethyl acetate. We can then prepare our aqueous phase, which consists of a 1% TPGS solution in water. After that, in the third step, we can add our organic phase dropwise to our aqueous phase. This causes self-assembly of our nanoparticles, as shown in the diagram. We then ensure that they're all the same size by using sonication before adding them to water and evaporating the ethyl acetate to remove the excess solvent. After that, we take our size measurements of our nanoparticles before centrifuging them. And when we centrifuge them, we can collect them as a macroscopic pellet as reflected in the diagram. Now moving on to the results section, in order to verify that our squalene molecule can bind to a ligand and that we can follow with the rest of our methodology, we take an NMR spectrum of the product produced at the end of squalination. I won't go into the details of how NMR works, but essentially the peak at about 11.8 ppm reflects that there is indeed a carboxylic group present in the squalene molecule. This lets us know that we will be able to proceed with the next steps of our methodology. Shifting to the formulation results, we're still in the early phases of experimenting with our squalene-naringenin or our squalene-ligand nanoparticle, but I will use the squalene nanoparticle control to explain the significance of the results and what we hope to observe when the study is complete. First and foremost is the size of the nanoparticles. Ideally, you want them to be between 200 and 350 nanometers. This ensures that they will be able to circulate through the body without causing any blockages in vital organs such as the kidney. Next is the zeta potential. This describes how stable our nanoparticles are. Specifically, the greater the absolute value of the zeta potential, the more stable the nanoparticle is, and the better it will be able to hold its shape. For reference, 10 millivolts, as shown here, is considered to be a relatively high value, so we want this value to be even higher for our squalene-naringenin nanoparticles. Lastly is the entrapment efficiency. This describes how much drug is encapsulated within the nanoparticle. As we can see, for the squalene nanoparticle control, this value is 20%, so we will want it to be much higher for our squalene-naringenin nanoparticle. Now moving on to the future steps. In the future, we're looking to optimize production of squalene-naringenin nanoparticles encapsulating paclitaxel. Again, paclitaxel is an anti-cancer drug, and this makes sense since the disease that we're trying to combat is cancer. We're also looking to do in vitro studies to evaluate drug uptake and cellular viability. Now at this point, I want to thank everyone for their time and ask if there are any questions.
Can you go back to slide 10 for me? So during this formulation phase, can you help us understand a little bit about what drives the self-assembly of the lipid nanoparticles?
Yes. So from my understanding, it's the hydrophobic effect, which basically describes how nonpolar substances want to minimize their contact with water. Paclitaxel and other class 4 drugs are very nonpolar, meaning that they want to avoid contact with water at all costs. Squalene, on the other hand, while being a mostly nonpolar molecule, the ligands give it some polar character. And so what happens whenever you mix the nonpolar drug, the squalene, and the water all together, the squalene will form a protective shell around the drug, which is paclitaxel in this case, to minimize its contact with water. And this also enables it to form the nanoparticle shape that we are looking for.
Hey, earlier you mentioned that lipid nanoparticles have become more popular since COVID. I was wondering if there was some reason for that, if it was related to COVID or something.
Yes. So during COVID-19, the mRNA vaccines actually use nanoparticles to deliver the mRNA to cells. This is because the lipid nanoparticles are made out of the same material as cell membranes, which allows them to easily diffuse through the cell and deliver the mRNA to where it needs to go. And so the FDA obviously approved these vaccines, and so that gave the green light for scientists, doctors, and medical researchers to begin using lipid nanoparticles to improve drug delivery.
Cool, thank you. And then I had one other thing. I was hoping you could elaborate on the concept of the off-site toxicity in regard to squalene.
Yes. So squalene, unfortunately, cannot differentiate between, let's say, liver cancer and areas of the body, other areas of the body that may exhibit cancerous conditions. And so this will cause the unmodified squalene nanoparticles to deliver the drug to other places besides its targeted route. This is why the ligands are really important, as they equip the nanoparticles with the ability to target specific protein receptors in the body that are in the area that we're trying to treat.